Now, we have been teasing our viewers because we've been talking about novel therapies, something in the pipeline, something good. So, Nusinersen.
Yeah, Nusinersen has been available since December of 2016 with a label for SMA. This is, again, an antisense oligonucleotide. It's intrathecally delivered, so requires lumbar puncture with both a loading phase and then a maintenance phase of dosing. And with the intent of this antisense oligonucleotide is it targets that SMN2 gene that all patients with SMA still have. Okay. And the SMN2 gene normally doesn't make SMN protein very well because of that nucleotide change compared to SMN1. But with Nusinersen on board, it alters the pre-messenger RNA to actually include an exon that's normally not part of the transcript so that the SMN2 gene can make SMN protein more efficiently and you get higher SMN protein levels in the patient.
Okay, so that was a genetic word salad.
Yes.
And I was following it.
Yeah.
You give this drug, you get more good stuff because the gene is made to work differently and better.
Yes.
I got that.
Yeah, you corrected your SMN deficiency.
That is, that's simply amazing. How did people come up with this?
I think, again, it's a genetically targeted treatment. You know, SMA is a highly appropriate target because there are these two genes that are so similar to each other. That's a very unusual situation genetically in humans. And we have this backup or homolog gene that is present in all patients who are affected that we can target. So it really was a unique target for an intervention such as this to come forward.
If there's such a thing as luck in this disease, having that backup gene is that. It's lucky. It's simply there and it just happens. But in order to know this, we had to do gene sequencing, we had to be able to do all that. We had to understand what was going on with the mechanism of this disease, why these people got these symptoms. That's a lot of heavy lifting.
And the pace is truly breathtaking. You know, we just understood the genetics of this in the mid-1990s, and then we were already in preclinical trials in the early 2000s with this technique, and we had the in the clinic trial starting in the 2010s, and we had an approved therapy in 2016. I mean, that's lightning fast in the context of drug development.
I'll put it to you this way, it's lightning fast, period.
Yes.
Because the complexity of this is appalling. And the fact that some of the gene sequencing is automated, I suspect, helps. But still, somebody had to think about this and do this. So now that this drug is out there, what is your personal experience with it?
So we have quite a large SMA center at Children's Hospital of Philadelphia where we have the majority of our patients on Nusinersen therapy at the current time because it's the only clinically available medication, although there are research trial alternatives that have been going on for a period of time now. And so our earliest patient was dosed in a couple of months after the approval, actually the next month after approval at CHOP in January of 2017.
What took you so long?
No,
a whole month?
That's right. Well, it was within weeks. And we were also part of the original research trials for Nusinersen, so we had patients on therapy for years before that in the research context. But we have experience across the severity types of SMA from a newborn baby up until an adult who's been living with SMA for decades, receiving this treatment, and lots of real-world data that we're starting to accumulate as a community and share in terms of what's happening in our patients with this treatment.
And what are you sharing? What are you seeing?
And what we're seeing are remarkable improvements in various age groups, various types of SMA. Certainly the peer-reviewed research publications about this, the pivotal trial, the ENDEAR study in the infantile onset and also the CHERISH study in type 2 SMA were both published in New England Journal of Medicine and show striking efficacy with high tolerability and remarkably minimal safety implications in terms of the medication.